Common structural features of transfer RNA molecules. The end of the chain of all tRNA molecules terminates with the 3 nucleotides C, C, and A. The amino acid which will be transferred is attached there, and there is an enzyme which attaches the correct amino acid to the transfer RNA, and that's where the proofreading in the system happens. It's the enzyme which is in charge of attaching the amino acid to the correct transfer RNA molecule. The cloverleaf model of transfer RNA has an anticodon loop consisting of 7 unpaired nucleotides. And 3 of those nucleotides will make up the anticodon, which will be complementary to the 3 codon bases of a messenger RNA. DNA is read 3 bases at a time. 3 bases form a codon, and then the codon codes for an amino acid. The other 2 loops in the cloverleaf structure enable the transfer RNA to bind to the ribosome and other specific enzymes during protein synthesis. So here we see the transfer RNA. There's 2 really important areas. The area, the CCA, where your amino acid is attached to, and then your anticodon, which this triplet here will be your anticodon. Transcription. This is the process of reading DNA and producing RNA with RNA polymerases, and it occurs in a complementary fashion and between appropriate bases on the DNA and the growing RNA. The genetic code. This is stored in the DNA in the sequence of bases, and again, 3 bases are a codon which code for an amino acid. In general, each codon specifies 1 amino acid. But the code is degenerate because there's 3 nucleotides. There's 4 possibilities, A, T, C, and G. And in 3 spots, that's 4 times 4 times 4. There's 64 codons for only 20 amino acids. And what you see is degeneracy. So there's 64 possible codes for 20 amino acids, so some have a lot of different codes. For example, here we see 4 codes. Here we see 4 different codes for serine. Here we see 4 different codes for leucine. And if we look further, we see 2 more codes for leucine. There's 6 codes for leucine. And so that's what we mean when we say the code is degenerate. There's 64 codes for 20 amino acids, so a lot of amino acids have more than 1 codon. Tryptophan only has 1 codon. And then there are 3 stop codons too. Methionine is always your start. AUG is always your start signal for translation. All right. And again, you read DNA 3 bases at a time is a codon. So if there are 200 codons, that's 600 nucleotides, because each codon is 3 nucleotides, and that will give you 200 amino acids for a 200 amino acid protein. The code from a gene in DNA is first transcribed to RNA, that's transcription, and then that messenger RNA is exported out of the nucleus via a nuclear pore, and then it lands on a ribosome, in which case it is turned into a protein, which is translation. So transcription is reading DNA and producing RNA. Translation is reading RNA and producing a protein. So the genetic code is universal for all living organisms. There's a couple exceptions when you're dealing with mitochondria and chloroplasts, but in general it's completely universal, which is one of the major pieces of evidence that makes us believe all life on this planet actually originated from one original cell. And the same nucleotide triplet specifies a given amino acid, regardless of whether it's in a bacterial cell, a pine tree, a human being. If you see AUG, it's going to be a methionine. So the sequence of nitrogenous bases on a strand of DNA is shown below. Indicate the nitrogenous bases of the complementary strand. Well, C binds G, adenine binds thymine, A binds T, C binds G, T binds A, T binds A, G binds C. The complementary to A is T. The complementary to T is A. The complementary to C is G. The complementary to G is C. And so if you have CACTTG, the complementary strand will be GTGAAC. So here we have another one. If we're transcribing, again, C becomes G. A, though, becomes U instead of T because RNA uses uracil instead of thymine. So back here it was thymine, but now since this is RNA, it's uracil. We're talking about this one right there. Again, C is complementary to G, T is complementary to A, T is complementary to A, G is complementary to C. So the only difference we see is from thymine to uracil because it's RNA instead of DNA. Oncogenes. These are genes that produce cancer. So they're present in cancerous or malignant cells, and they code for proteins that produce uncontrolled cell growth. Viruses are very good at inserting oncogenes, and so there's a lot of human cancers which are caused by viruses. So HPV causes cervical cancer and head and neck cancers. There's 2 human leukemia-causing viruses. There's a lot of viruses that cause cancers because they can insert oncogenes into a cell and rapidly transform it into a cancer cell. Tumor suppressor genes are the genes that produce products like P53, which actively block cancer development. And if a cell becomes potentially cancerous, a cell will destroy itself. So apoptosis is a regulated, orderly cellular suicide in which a cell destroys itself for the good of the whole entity because the cell thinks it's becoming cancerous. The Human Genome Project started in 1988 to determine the complete sequence of the human genome, which is 3 billion base pairs long. And by 2001, the complete sequence was known. And, back then we thought there was about 23,000 protein coding genes. That's been updated. We think there's 30,000 protein coding genes. And actually in the newest research, when they're looking for transcription start points, they find 160,000 transcription start points. And so the newest theories in molecular biology are thinking that maybe there's 30,000 or so protein coding genes in the genome, but there's probably like 130,000 RNA producing genes in the genome, of which we know almost nothing about. And so there is a lot to learn about of the human genome before we can say we understand it at all. The vast majority of the human genome has an unknown function or no function at all. It used to be called junk DNA because no one knew what it did. But in reality, we were realizing more and more that what we used to call junk DNA is actually super important. We just didn't understand what it did. Gene therapies. A major goal of the Human Genome Project is to develop gene therapies for inherited diseases. And progress has been slow, but there has been some recent successes. A fatal immunodeficiency caused by a deficiency of adenine deaminase and the serious immunodeficiency made famous by the boy in the bubble have both been cured using gene therapy. And so we are slowly developing gene therapies for more and more genetic diseases. Approximately how many base pairs in the human genome code for proteins? About 1.5% of your actual genome codes for proteins, so very little. And so again, 1.5% codes for proteins. A lot more of the genome, up to 20-25% of the genome is in genes. But in your average human gene, only 4% of the gene actually is made into proteins, and the rest are introns, which do not become amino acids. Translation. This is the production of a protein. A ribosome reads in a messenger RNA, transfer RNAs come in, and the ribosomal RNAs of the ribosome connect the amino acids into a polypeptide. So the function of transfer RNA is to bring amino acids to the protein synthesis site on the ribosome. And translation is broken down into 3 stages: initiation, elongation, and termination. So there's 3 spots in the ribosome we're going to look at.